A very good morning to all of you. Today we will study about different type of antibodies, or you can say different type of immunoglobulin. So when we are dealing with the different type of antibodies or the immunoglobulin, the first antibody which comes is IgG, that is antibody G or immunoglobulin G. When we are dealing with IgG, it is a antibody whose structure is monomer. Monomer means it is having only a Y-shaped structure. Now the second thing is that this is the antibody which is present in maximum amount in our body, in our serum. So the percentage of serum antibodies means the concentration of this particular antibody, that is IgG, its amount in the serum is 80%. So it is the maximum antibody, maximum amount of antibody which is present in our serum. Now comes where we can find, where we can isolate this particular antibody. So first of all, we can isolate this particular antibody from the blood, then from the lymph, and then from the intestine. In the same manner, when we move on, this antibody, it has a high half-life in the serum is 23 days. Now this particular antibody, it is the antibody which shows complement fixation, and this is the only antibody which cross the placenta, means this is the antibody which we get from our mother. So this is the only antibody which cross the placenta. And now comes the main functions of this particular antibody. The main function of immunoglobulin G or antibody G is that it enhances phagocytosis, means it speed up the process of phagocytosis. In the same manner, the second main important function of IgG is that it neutralizes the toxins and the viruses. Different type of bacteria, when they invade our body, they will give rise to different type of toxins. So the main function of this immunoglobulin G is that it neutralizes the toxins and the different type of viruses which are invading our body. And the last but not the least, or the main important character of IgG or immunoglobulin G is that this protect fetus and the newborn. This is the antibody which protect a fetus, which protect a newborn baby until and unless his immune system starts working. So this is all about IgG. Now we move on the second antibody, and that is IgM, immunoglobulin M, or the antibody M. When we are dealing with antibody M, its structure is pentamer. This is a very different type of antibody. It is having 5 type of Y-shaped structure. That is why its name is pentamer. Now when we are dealing with this particular antibody, the percentage in the serum is 5 to 10%. So when we are dealing with this particular antibody, the amount of this particular antibody, it is less in the serum. And from where we can isolate it? We can isolate this from the blood, from the lymph, and from the B cell surface. So its half-life in the serum is 5 days only, and it also shows complement fixation. This antibody, it will not cross the placenta, and the main function of this immunoglobulin is that this is the first antibody produced during an infection. So this particular antibody, if it is present in our serum, it gives an indication that our body is suffering from an infection, or our body has been invaded by a bacteria or a virus. Now it is this antibody, that is this pentamer, this IgM antibody, it is effective against microbe and agglutinating antigens. So when we are dealing with this particular IgM, so main character which you should learn from this, that the main characteristic is that it is a pentamer, it is having 5 Y-shaped structure. Its main function is that, or its location is from the blood and the lymph for the B cell phase. And in the same manner, this is the particular antibody which will be first appear in the blood when we are having any type of infection. Now we move on the next, that is immunoglobulin A or the antibody A. When we are dealing with this antibody A, its structure is dimer. Dimer means it is having 2 Y-shaped structure. Its percentage in the serum is 10 to 15%, and its location is in the secretions. So we are having different type of secretion in our body. We have tears as a secretion, we have saliva as a secretion, intestinal layer, and the milk and blood and lymph. These are the particular locations from where we can isolate immunoglobulin A or antibody A. Its half-life in the serum is 6 days, and it do not fix the complement. It also do not cross the placenta, and the main function of this particular immunoglobulin A or as you can say, this is also known as secretory antibody. So secretory antibody main function is that it is localized protection of mucosal surfaces. It provides immunity to infant digestive tract. So when we are dealing with immunoglobulin A, so this is present in the secretion, this is present in the human milk, it is present in the intestine, it is present in the saliva, it is present in the tears. So this is the antibody. As IgG is going to protect the antibody, protect the fetus, in the same manner this IgA, it is having a dimer structure, so it is also helpful in protecting the fetus from different type of infections. Now we come on the different antibody, and that is IgD. When we are dealing with IgD, its structure is monomer. Monomer means only a single Y-shaped structure. Now its percentage in the serum is very less, that is 0.2%, and its location is on the B cell surface, that is IgD. It is present on the surface of the B cells, blood and the lymph. Its half-life in the serum is 3 days only. It do not fix the complement, it do not cross the placenta, and its known function are in serum its function is unknown, but on the B cell surface it initiates the immune response. So this is the main function, that the B cells, when they are behaving, or when they are responsible for the immune response, behind that B cell activation IgD also play a important role. Now we come on the IgE. This particular antibody, its structure is also monomer. Its percentage in the serum is only 0.02%, and its location is, it is bound to the mast cells and the basophils, and it is throughout the blood, throughout the body, and in the blood also. And half-life in the serum is only 2 days. It also do not cross the placenta, and it do not fix the complement, and its known function is, it is appear during an allergic reaction and possibly lysis of the worms. So if we are infected with any parasite at that time, this IgE, it is helpful for our body, it will kill that particular worms. So this is all about the different type of antibodies which we are having. So give a quick recall these particular antibodies. First is IgM, then this is IgA, then IgD, IgE. So these are the different type of antibodies which are behaving a important factor which protect us from infections.